The overall goal of this screening assay is to characterize novel endothelial regulators involved in the inflammatory response of interest for designing new therapeutic strategies against inflammatory disorders.
This method can help answer key questions in the pathology field, such as defining novel endothelial targeting drugs able to attenuate inflammatory-related diseases. The main advantage of this endothelial protocol is that it describes a stepwise technology that establishes the basis for exploring novel mechanisms involved in the regulation of the inflammatory response. Demonstrating this methodology will be María Ángeles Higueras, a postdoc from our lab, and Sandra Herranz, a technician from our lab.
To begin, culture MLEC-04 cells in complete DMEM F12 medium supplemented with 10% FBS and penstrep. Incubate the cells in a biological incubator at 37 degrees Celsius with 95% humidity and 5% carbon dioxide. Coat 100 mm tissue culture plates with 2.5 milliliters of gelatin solution and incubate the plates at 37 degrees Celsius for 30 minutes. Aspirate the gelatin solution and leave plates in the tissue culture hood to air dry. When the MLEC-04 cells reach confluence, split them 1 to 3 by first using 10 milliliters of sterile PBS to wash the plates. Then add 2 milliliters of trypsin EDTA. And incubate the cells at 30.